Greetings everyone. So today we are going to discuss about a new topic which is asexual reproduction. Uh, Basically, we are done with the growth of bacteria. That how actually bacteria have the mechanism of growth within them. Uh, Here we are going to discuss about the reproduction overall and first part of reproduction, which is the asexual reproduction. So in bacteria, there are two types of reproduction. First includes the asexual reproduction, in which there is no fusion of gametes, there is no fusion of male and female gametes. But in case of another type of um, reproduction, we are not having a sexual reproduction, but rather than a process which is similar to sexual reproduction, which is what we call as parasexuality, which is known as parasexuality. Parasexuality, and in other words, we can also call it as genetic recombination, and that basically involves the transfer of genetic material from one bacterium to other bacterium, which occurs in three ways, which includes transduction, transformation, and conjugation. We'll discuss that in a separate video. For now, we are going to focus on the asexual method, which is uh, what we call as asexual reproduction, in which we are having two conditions. First is favorable condition, and second is the unfavorable condition. Unfavorable condition. In case of favorable condition, we are having two processes which includes budding and binary fission. Budding, binary fission. In case of unfavorable conditions, we are having two processes which includes cyst formation, which is known as encystment, which we have previously studied, and after that we are having formation of exospore formation. Now in this case, these are the processes which we have previously studied in cyst formation and spore formation. Here we are going to discuss about budding and binary fission. So when it comes to the process of budding, uh, before discussing budding, let us just have a look into the process of binary fission. So when it comes to binary fission is a most common means of reproduction, and particularly asexual reproduction. uh, when it comes to asexual reproduction, it is the most common reproduction occurring. Parasexuality occurs, but it occurs quite rarely. Apart from which, when it comes to asexual reproduction, particularly, we are having binary fission as the commonest method of reproduction and occurs nearly in all bacteria. After that, when it comes to the process, how does it actually occur? It basically is also known as amitosis, because that occurs, uh, you know it is known as amitosis, which means it is a process which is, even not same as meiosis nor mitosis, and thus we call it as amitosis, because it is a separate process which specifically uh, associates with prokaryotes, such that it is the only process which is occurring in prokaryotes. However, in eukaryotes, the process of binary fission occurs in as well. In case of abnormality, binary fission occurs in eukaryotes as well, such as yeast, as well as in other eukaryotes. The process of binary fission occurs, but in case of abnormality. Here in prokaryotes, it is a regular process, and that is known as amitosis. When it comes to its structure, um sorry, the mechanism occurs in such a manner that the first step involves the replication of the nuclear material, genomic material, due to which the replication of the nucleoid occurs, such that after the replication of nucleoid, the replicated nucleoid material has now turned itself from haploid to diploid, which means this is a stage of cell division where the genetic material has been converted, has been replicated from haploid to diploid, such that here the diploid material is being formed, right? And that diploid material is formed only for a short period of time, after which the nuclear material which is replicated now moves towards the opposite poles. Now here the movement of the nuclear material towards the opposite poles will not take place due to cyclosis, because there is no cytoskeleton present over here. but the process will take place due to the presence of several cytoskeletal materials within the cytoplasm. Now, cytoskeletal material is present within the cytoplasm, which facilitates the process of movement of the nucleoid towards the opposite poles, as a result of which the cytoskeletal material leads to the movement towards the opposite pole of the nucleoids. Once the nuclear material is then transferred towards the opposite pole, the third step involves the formation of a cleavage fluid, in which there is, formation of a transverse septum, formation of a transverse septum. Let's suppose if we take the example of the bacterial cell. Let me just try to draw the structure. So there will be formation of a transverse horizontal. We are having two sections, longitudinal section and transverse section. So there will be formation of a transverse septum, and that transverse septum form will cause to separate the cytoplasm of the bacterial cell, as a result of which the bacterial cell will now divide into two separate daughter cells after the formation of the cross wall or the septum. After that, this is the process of binary fission. Usually the process of binary fission requires 20 minutes, 20 to 30 minutes, but in E. coli bacteria it requires around 18 minutes. After that, when it comes to budding, budding is a process which occurs uh, in special bacteria species which are known as Planktacea. These are basically aquatic bacteria which live in aquatic environment, and these are the bacteria where the process of budding occurs mostly. And in this case, we are having the process occurs in such a manner that here there is a formation of bud. Bud is basically an outgrowth of the cell, such that uh, in this process, outgrowth of a cell is formed which separately grows into another organism. That process also occurs in eukaryotes such as yeast, as well as in some other eukaryotes such as animals as well. But in case of, sorry, it's the budding process in bacteria. So here we can better write it as bacteria. Anyways, the process of budding would be quite similar of yeast with bacteria, which we are going to discuss here. Such that first there would be formation of an outgrowth of cytoplasm. There will be no formation of outgrowth of nucleoid, only outgrowth of cytoplasm would be there. Uh, let me just draw another diagram. So let's suppose this is a cell, and that cell now basically the first step causes to form an of outgrowth of the cytoplasm. This is a nucleoid material as well. Nucleoid material. There is no outgrowth of nucleoid material, only the outgrowth of cytoplasm. After which the nucleoid material replicates inside the cell. nucleoid material replicates Nucleoid material replicates, right? It is trying to draw a rough structure, and here as the nuclear material replicates, the cell again becomes diploid. So there's a diploid stage occurring, after which as the nuclear material is replicated, there will be, let me just try to draw the structure again. So here as the nuclear material is replicated, now the cell membrane and cell wall will now tend to constrict. Cell wall and cell membrane will now tend to constrict. After replication, the cell wall and cell membrane will now tend to constrict. As a result of the constriction will occur from this part. Constriction of the cell wall and cell membrane occur, due to which the outgrowth will be now forming a separate bud. Will be now forming a separate bud. Once constriction occurs, there is formation of a separate bud. There is formation after the constriction. There will be formation of a separate bud. Now, after that, after the formation of the bud, there will be the separation of the bud. Separation of the bud will be there, right? And after separation of the bud, After the separation of the bud, we'll be basically having a separate bud which will mature itself, such that when it will mature itself, it will remain fully functional to grow itself as a separate bacterial cell. After the maturation, it would be developed into a separate bacterial cell, right? So this is how the process of budding will occur, and um this process of budding is just that simple. After that, when it comes to another point which I would like to highlight in the binary fission, that after the formation of the septum, the cell wall which is formed outside, the cell wall which is formed outside will be degraded, such that after the formation of the septum, the outer cell wall, which is older cell wall of the parent cell, will degrade, as a result of which a new cell wall will be formed of the two individual daughter cells, due to which two individual daughter cells will have a separate daughter cell wall in the new daughter cells. So this is all about the process, apart from which we are going to discuss about something else in the next video. Hopefully we'll meet in another video.